it's really a thrill to be here and to imagine that it's been 50 years, but it has been. So what I'm going to try to do is give you some sense of how this all began. And first we have to kind of set the scene. The PDB did not just pop up. And to think about it, we have to first think about the pioneers of structural biology. The first being J.D. Bernal at Cambridge, who was called the Sage for good reason. He and Dorothy Hodgkin in 1935 got the first diffraction photograph of a protein crystal, which was really remarkable. And more than 20 years later, we finally actually saw the first crystal structure of a protein, myoglobin, done by John Kendrew and colleagues, followed very shortly thereafter by the hemoglobin crystal structure. Here are 4 pictures drawn in different representations of these landmark structures: myoglobin from the Kendrew lab, hemoglobin from the Perutz lab, lysozyme from David Phillips's lab at Oxford, and then ribonuclease, which was in 1967 and was the very first American protein crystal structure. In the same era, Chris Anfredson studied protein folding in vivo, and through his work, we understood that the sequence of proteins determined the structure in ways that we still don't quite understand, but his seminal contribution, which won him a Nobel Prize, was very important and motivating for people who cared about protein structure. There was also significant effort in silico. Another pioneer, Cyrus Leventhal, studied the folding of proteins computationally. He was originally on faculty at Michigan, but moved to MIT, where he was heavily involved in Project Mac, which was an attempt to visualize and understand proteins. And I show on the right a picture of a postdoc of his at the time, Edgar Meyer, in front of the very first computer graphics for protein structures. And then we need to acknowledge greatly the pioneering work of Olga Kennard, shown here on the left, who founded the Cambridge Crystallographic Data Center, which was the archive for small molecule crystallography and very much influenced the development of the PDB. Then there was, remember, this was the 60s, and there was a lot of community activism, people who really cared about these protein structures and wanted to have a way to look at them. And if you think about those times, that was a difficult task. We had punch cards, a little bit later, magnetic tape. How would you ship all this around the world? And so 3 of us were particularly active at that period. We used to write letters and talk on the phone about how we can make a protein data bank. And that was Edgar, Gerson Cohn, who at the time was at NIH. He's both Edgar and Gerson are no longer with us, and myself. We had lots of meetings. And interestingly, the American Crystallographic Association took a very, very active and supportive role in helping us form the idea of a protein data bank. On the right, I show you some a petition that we wrote in 1971 in February. And you'll see some names here that are at this meeting. Shoshana Vodak and Joel Sussman were very much involved. Donald Vogt, and so on. And these are all people who really thought this should happen, but of course had no idea how it would happen. So 1971 was a turning point year. I told you we had that petition in 71 in the winter. And then in June of 1971, there was a meeting at Cold Spring Harbor on the structure and function of proteins in 3D. And I was, at the time, I came to that meeting with Joel and Shoshana, I believe. And we were just, it was thrilling to see all these people who had done the very first protein structures talking about their work, and it was really inspirational. At that meeting, those of you who've ever been to Cold Spring Harbor, there's a bar near the meeting site, the meeting room, and we were all hanging out there. And Walter Hamilton, who had been in the chemistry department at Brookhaven, and at the time was working on the structures of amino acids, was at that bar. We approached him and we said, we really need to have a PDB. We have to have a PDB in the way that people in the 60s talked. And he said, fine, we'll have a PDB. And so he flew over to England, and he wisely met with Olga Kennard, because he knew if we were going to have a PDB, that it had to be a global enterprise. And Olga agreed to collaborate. Edgar had already been working on creating the technology for the PDB based on the work he had done at MIT. He was working during the summers at Brookhaven. And then they invited me to become what we now call an annotator. And that meant that I had to punch coordinates up on a key punch. And in the October of 71, there was an article in what was called at the time Nature Biology, in which the PDB was announced as a collaboration with Europe. And that's how it all really began. And it was a lot of grassroots effort, and then a lot of real important passion and expertise that led to the PDB. So once we had the PDB, in principle, please remember there may be, well, the official number we say is 7 structures, although we'd have to figure out how we did that count, but it was a very, very, very few structures. Began at Brookhaven. There was no funding, of course, but there was another project that had been formed at Brookhaven by Walter and Edgar, called CRISNet. And that was a remarkable project because at the time, the idea was to have graphics terminals distributed in labs around the country, and the main computing be done at Brookhaven. At the time, it was called the CDC 6600. And Edgar and Walter invited me to be setting up one of those labs. At the time, I was in Philadelphia at the Fox Chase Cancer Center. That grant received its funding. The funding letter we received was on the day of Walter's funeral. Walter passed away at a very young age in 1973, and asked Tom to head up the PDB, which he did. A few years later, we found a picture, Tom Ketzel found a picture of the PDB team with the 100th crystal structure in 1979. And you'll see in this picture, Fran Bernstein, who we brought on as what we now call an annotator or a biocurator to handle the structures. The Graham Williams, shown on the left, was involved with the CRISNet project, and Tom Ketzel was leading that. And those were the very beginnings. In 1994, Joel Sussman took the lead of the PDB at Brookhaven, and I show you on the right a clear color picture of the staff of the PDB at Brookhaven. In 1999, the management of the PDB changed, to the Research Collaboratory of Structural Bioinformatics, RCSB. And that was a consortium consisting of Rutgers, San Diego Supercomputer Center under Phil Bourne and Peter Osberger, and NIST under Gary Gilliland. And on the left is a picture from the NSF when we were kind of given our marching orders as to what we had to do. And as you might imagine, after all those years of successful operation at Brookhaven, we had our work cut out for us, and we took it very seriously. I show you in the middle picture, the staff was very devoted to the PDB, and there was a big Hurricane Floyd, which flooded all the streets in New Jersey, and we weren't supposed to go anywhere. But of course, the gang showed up. There was no water, hence the porta potties, in order to process data. And then on the right, I show a picture of a more modern picture of the current of the PDB staff. At the same time as the RCSB received the contract, there was work going on at EBI and in Japan to have data centers. And the one at EBI was called MSD, now called PDBE, and the one in Japan is called PDBJ. We decided, all of us, that it was really important that we have a single archive and that things didn't get split up. So that's how we formed the worldwide PDB. The agreement for the worldwide PDB is that we would make sure that there was a freely and globally available archive and that our data processing and annotation procedures would be the same, but that our websites could provide different kinds of services. And that is what we did. In this picture, I show you, we had an international advisory group. I show you one example of the many that have meetings that we have had. And you can see Janet at this particular meeting that I show here. And on the right was the very beginnings of trying to make a single deposition system, no matter where in the world it was going to be. And this was a meeting that the WW had in order to make that happen. And this shows you the people who were doing that at that time. So how do policies get set up? From the beginning, as I've told you, there's been very active community involvement. The most important policy thing that happened was to establish deposition guidelines. There was no imperative to put data in the PDB, but many people felt that it was essential that the data, that it become mandatory for the data to be in the PDB. And various committees were set up, including by the IUCR, to figure out what that really means. There were also a very active involvement by the journals and discussions with the journals, and the funding agencies were enormously encouraging in this effort. On the next slide, I show you the kind of things that happened. There were people at the time who were extremely unhappy about the data not being mandatory, and there was an article in Science about that subject, and a remarkable petition that was headed up by Fred Richards and brought in some of the key protein crystallographers at the time, asking them to sign this petition. And I believe there were 200 names on that petition. And then based on that, it was possible to make the deposition mandatory. The other issue was the creation of data standards. The PDB had a format called the PDB format, but many felt that there needed to be a more complete format, and a group came together, sponsored by IUCR, to work on it. It was headed by Paula Fitzgerald, shown here on the top picture. And this is a meeting that Shoshana Vodak hosted in Brussels, where we really set the plans for making the MMSIF. And on the other people, a key person was John Westbrook, who was really the architect of MMSIF, and you can see Phil Bourne, who's on the committee, and Sid Hall, who was the architect of small molecule SIF. We had the first draft in 1996, but it was not adapted as the master format until 2011. The other very, very important issue was validation. How do you know that a structure is correct? In 2008, we formed, the WWPDB formed an X-ray validation task force headed up by Randy Reed, bringing together experts in validation to figure out exactly how structures should be checked. And by that time, structure factors were mandatory in the PDB, and therefore it was actually possible to really check the structures. Then there was the NMR task force, the EM task force, small molecule small angle scattering, and then very recently the integrative methods workshop. All of these groups worked to form the standards and to write white papers, which were the basis of the validation, are the basis of validation by the PDB. What I think I want to emphasize here is that it's not just about science, it's not just about technology, it's not just about community. All of these aspects have to work together in order to make the PDB a living thing that is meeting the needs of the time. So I'll just, for those of you in the audience who've never seen a punch card, right over here in the 1970s, this is the way data was managed on these punch cards. And the technology kept changing, science changed with it, community kept changing, and that kind of change is very important. Today, there are more than 175,000 structures in the PDB, 15,000 new depositions in 2020. So if you think about, we started with a few, 7. And as Celia pointed out, 2.4 million data files downloaded every day. Here's a picture of the structures in the PDB organized by function, which was drawn by David Gutzel and Maria Voigt, and then a very impressive way of showing you the diversity in the PDB. Along in time, there were celebrations. There was a celebration of PDB 25 in 1996 that Joel organized in Jerusalem in collaboration with Amos Bayrak, celebrating the 10th anniversary of SwissProt. And then in 2011, we had the big celebration for PDB 40, and we chose to have that at Cold Spring Harbor to honor the birthplace of this whole concept of the PDB. And before I end, when I tried to make the acknowledgement slide, I realized that there were just too many people. So please forgive me for not naming everybody, because it's just impossible. But I think everybody here at this meeting and all the people who've deposited data, who've analyzed the data, who've developed drugs from the data, the teachers and students, the journals, the funding agencies, and the activists. I told you about the early activists, but there are a significant number of people who have kept a close eye on the PDB and are never shy in saying how things should change and what things should happen. And then finally, I want to make a special tribute to some very important people who made the PDB happen. First, of course, Walter Hamilton, shown on the left, Edgar Meyer, who was responsible for the early technology, Fred Richards, who took a strong role in galvanizing the people in the community, Michael Rossman, of course, John Woolley, who kept us on our toes. He worked with DOE and with NSF. And then finally, Marvin Kastman from NIH, who made sure we understood how important it was to keep the data public. And with that, I would like to thank you for your attention.
Helen, thank you very much indeed. I think we ought to have a virtual clap for a beautiful summary of what happened in the beginning. I can still remember as a young postdoc getting the data tapes, the 12-inch magnetic tapes with the PDB files. I think there were 20 on it the first time I got a data tape. But I think if I could just ask you, if people have questions, please post them in the chat. But I'd just like to ask you, Helen, we've heard about how things started. Where do you think, what will the PDB look like in another 10 years even? I won't ask you 50 years, that's too hard, but maybe 10 years.
That's a very interesting question. Well, I think we're going to see more and more of these macromolecular machines. We're going to see a lot of integrative structures. And as you know, we're busy working on figuring out how to actually do that. I think at some point the PDB has to make a boundary, and hopefully there will be other groups who will take the lessons that from what we did, and hopefully there'll be more databases having to do with the cell and beyond. Okay, and so I think at some point all of us here have to make this figure out what that boundary is. The structures, one thing that we recently found out is that EM is going to surpass X-ray as the method of choice. But that's now, and we don't know what methods are going to be out there because, and biophysics will continue to the biophysical community will continue to cooperate, and we'll figure out how to bring all of this together.
Okay, there's one question related in the chat. Asking, how do you think will structures from in-cell tomography integrate with the PDB? So how are we going to put it all together?
Well, I think we have to, we're not going to, I don't think we can, well, this is just my view. Okay.
Yeah, of course.
We're not going, I don't think we should put it all together. I think at some point we have to form partnerships, and we really need, and you know better than anybody, how important it is for all the different data resources to truly cooperate, okay? And then I think we could have these really important partnerships, and it's beginning to happen. It's beginning to happen.
Yeah, thank you.